Assalamu alaikum wa rahmatullah. Hello everyone. Our group today will discuss quality control of liquid dosage form as a part of our pharmaceutical technology course. These are our content outlines we're going to discuss today, starting with a simple introduction, moving to types of liquid dosage form and general test and raw materials, moving to emulsion and suspension tests, ending with additional tests that may be carried to for quality control reasons. First of all, we need to know what does liquid dosage form mean. Liquid dosage forms are portable pharmaceutical formulation composed of a blend of active component and excipients that are dissolved or suspended in an appropriate solvent. We have two major reasons why we make our drug in liquid dosage form. The first one for the patient whom facing difficulties in swallowing solid medication, or for the drugs need a fast-acting therapeutic effect. Secondly, we need to know what's the importance of quality control to be implemented in manufacturing liquid dosage form. Quality control is implemented in manufacturing of liquid dosage forms for several important reasons. Here are some reasons. Starting with the patient safety. As we all know, the drug manufacturing is one of the most critical industries around the world because it's concerned the human health. Secondly, the consistency and the predictability. Thirdly, the regulatory compliance. As here in Saudi Arabia, we have the Saudi FDA. It's the authority who's responsible for the regulatory affair. Fourthly, the patch-to-patch uniformity. We are manufacturing millions and millions of the drug. All should be the same, all should have the same criteria. Fifth and last, the repetition and trust. Every company seeking for this point. Here we have like a simple classification for the liquid dosage form. We can classify it as a monophasic or biphasic. Then we have subdivision under each. Monophasic can be divided into aqueous and non-aqueous. For the biphasic, it can be divided into suspensions and emulsions.
Type of liquid dosage form. Oral solution. Oral solution are oral liquids containing one or more active ingredient and excipient, dissolved in suitable fecal. Water is the most common solvent, although organic solvents are used in combination with water or on the own. All the components of solution are dispersed as molecules or ions, and the solution is optically clear. Oral suspension. Oral liquid containing one or more active ingredients suspended in a suitable fecal. Suspended salt may slowly separate on keeping but are easily re-separated. In the manufacture of oral suspension containing dispersed particles, measures should be taken to ensure a suitable and controlled particle size with regard to the intended use of the product. Oral emulsion. Oral emulsion are dispersion of at least two miscible or partially miscible liquid. There are oral liquids containing one or more active ingredient and are stabilized all in water dispersion, either or both phase of which may contain dissolved salt. Salt may also be suspended in oral emulsion. Emulsion may exhibit phase separation but are easily reformed on shaking. General test of liquid dosage form. Visual inspection. Oral solution and oral drop should be clear and free from precipitate. Discoloration or cloudiness of a solution may indicate chemical degradation or microbial contamination. The fitness of physical instability of oral suspension and oral drop. There are suspension is demonstrated by the formulation of fluoricant or sediment. There are don't ready despair on gentle shaking. In case of oral emulsion and oral drop, that oral emulsion fitness of physical instability is demonstrated by phase separation that is not readily referred on gentle shaking. pH test. Litmus paper is a common pH test used to determine the acidity of or basicity in solution. This is common in the three types red, blue, and neutral. For a specific pH result, a more precise pH test paper or strip is needed. This strip and paper are quick, easy to use, less expensive, portable, and suitable for field work. However, they are inferior to pH meter and may not provide the best result due to color and turbidity concern. pH meter are process tests that measure the solution pH by measuring the electrical potential difference between the pH electrode and a reference electrode. They are useful for fitness scientists, college, or research work. Handheld portable pH meter are ideal for field work or lab check. Bench pH meter offer more specification and more testing options. Raw material specification and test. Raw material specification are crucial in liquid product formulation due to contaminated and the inclusion of sweeteners and thickness agent. Water is the most common raw material and manufactured most completely with purified standard. Oral solution containing active ingredient and excipient with the water being the most common solvent. Raw material and grow rigorous tests for purity, identity, and quality before manufacturing depend on the product type. 15, 20, 16 materials may be needed, with the manufacturing decimation testing extent.
Test for suspension. We have 3 tests for suspension. First, the sedimentation volume. Second, the redispersibility test. Third, the particle size change. Sedimentation volume test. The sedimentation is measured as the final sediment volume, which is VO, relative to the starting suspension volume, VO. And this relationship is denoted by the ratio F, where F equals to VU divided by VO. If the value of F is equal to 1, that means there is no sedimentation and no clear supernatant. If the value equals to 0.5, that means 50% of the total volume is occupied by sediment. If the value is greater than 1, then the sediment volume is greater than the original volume due to the formation of floccules which are fluffy and loose. The redispersibility test refers to how easily the sediment that has formed can be mixed back into the suspension to create a homogeneous mixture with moderate shaking. The suspension's redispersibility can be assessed in 2 ways, either by gently agitating the suspension in its container or by the other method, which is by employing a mechanical shaking device that mimics the motion of a human arm during shaking. This method provides more consistent and reproducible results. Particle size change test. Any change in the particle size within the suspension can be an indicator for a potential crystal growth. To test this, we use a freeze-thaw cycling technique and measure the particle size with either a microscope or a culture counter device. Note that before the measuring, we dilute and break apart the suspension to ensure we are measuring individual particles rather than clumps. This method helps us predict how the suspension will behave after long storage at room temperature. Test for emulsions. Also, we have 3 tests for the emulsion. First, the phase separation test. Second, the droplet size. Third, dilution test. The phase separation test, which is visual assessment, provides an approximate estimation of the phase separation. Usually, creaming, flocculation, and calcins can occur before the phase separation becomes visible. This can sometimes complicate quantitative evaluation. The rate and extent of phase separation in an emulsion can be readily determined by retaining a specific quantity in a graduated cylinder and measuring the volume of the separated phases at defined time intervals. Phase separation may arise from creaming or coalescence of globules. To accelerate the phase separation, low or mild rate centrifugation can be employed. Droplet size test. Any increase in droplet size after making an emulsion suggests that the emulsion is becoming unstable. Droplet size can be determined using a microscopic methods or electronic devices such as a cutter counter device. For emulsions with droplets larger than 1 micrometer, optical microscopy is especially useful because it provides a direct and reliable measurements of individual particle size. This is an example of a cutter counter device just to imagine how it works and its mechanism. As we can see, the particles enter the cathode and is recorded in the screen. The second particle, we see the second wave. The dilution test. is used to determine the type of an emulsion. Look at the picture down the screen to imagine the process. We have a test tube, adding inside the test tube a few drops of emulsion, then add a few drops of water. We have two situations. The first situation where the water distributes uniformly, that we will know from the emulsion type is oil in water. If the second situation, if the water separates outlier, we know then the emulsion type is water in oil.
Additional tests. First, uniformity of content test. For this test, as per British Pharmacopoeia assay, we select 10 units individually using an appropriate analytical method. Then we carry out the assay on the amount of well-mixed material that is removed from an individual container in condition of normal use. We calculate the acceptance value using the following formula: m minus x plus k multiplied by s, where m is the reference value, m is the mean of individual content, and we express it as percentage of the label claim, k is acceptability constant, and s is the sample standard deviation. According to British Pharmacopoeia, the preparation complies with the test if not more than one individual content is outside the limit of 85% to 115% of the average content, and none is outside the limit of 75% to 125% of the average content. The preparation fails to comply with the test if more than 3 individual content are outside the limit of 85% to 115% of the average content, or if one or more individual content outside the limit of 75% to 115% to 125% of the average content. Second, uniformity of mass test. For this test, we weigh individually the content of 20 dosage unit taken at random, emptied as completely as possible, and determine the average mass. As stated by British Pharmacopoeia, for single dose preparation that are solution or emulsion, not more than 2 of the individual masses deviate by more than 10% from the average mass, and none deviate by more than 20%. For single dose powders and granules for syrups, oral solutions, oral suspensions, and single dose powders for oral drops, according to British Pharmacopoeia, not more than 2 of the individual masses deviate from the average mass by more than 10% deviation if the average mass is less than 300 mg, or 7.5% deviation if the average mass is 300 mg or more. Third, uniformity of volume test. According to British Pharmacopoeia, this test is suitable for oral liquid and oral suspensions of viscous preparations. For this test, we select a sample of 10 filled containers and determine the weight of the content of each container. Determine the weight per milli and calculate the net volume of the content of each container. For non-viscous and free-flowing liquid, we pour completely the content of each container into calibrated volume measure of the appropriate size and determine the net volume of the content of the 10 containers. Consistent with British Pharmacopoeia, the average net volume of the content of the 10 containers is not less than the labeled amount, and the net volume of the content of any single container is not less than 9% deviation if the net volume is 50 ml or less, and 4.5% deviation if the net volume is more than 50 ml but not more than 200 ml, and not less than 3% deviation if the net volume is more than 200 ml but not more than 300 ml. 4. Uniformity of weight test. For this test, we select a sample of tin-filled container and remove any labeling that might be altered in way while removing the content of the containers. We clean and dry the outer surface of the container and weigh each container, remove quantitatively the content from each container, and if necessary we cut open the container and wash each empty container with a suitable solvent, taking care to ensure that the closure and other part of the container are retained. We dry and weigh each empty container together with its part which may have been removed. The difference between the two weight is the net weight of the content of the container. As per British Pharmacopoeia, the average net weight of the content of the tin container is not less than the labeled amount, and the net weight of the content of any single container is not less than 9% deviation if the net weight is 50 gram or less, and not less than 4.5% deviation if the net weight is more than 50 gram but not more than 100 gram. In the end, I would like to summarize what we have talked about in this video. We have talked about quality control test of liquid dosage form, which can be classified to unofficial test and official test. For the unofficial test, we use visual inspection for solution, and for suspension we use sedimentation volume, redispersibility and particle size change. For emulsion we use phase separation, droplet size and dilution test. As for the official test, examples are uniformity of content, uniformity of mass, uniformity of volume and uniformity of weight. And thank you for your attention.